I'm Daniel Levine, and this is the Bio Report. The ARC Virtual Cell Atlas uses high-throughput single-cell genomics, artificial intelligence, and open science to understand the complexities of cellular behavior. Developed through a partnership between the ARC Institute, 10x Genomics, and Ultima Genomics, the public domain resource integrates data from hundreds of millions of cells. By curating and harmonizing vast amounts of single-cell measurements, the Atlas paves the way for virtual cell simulations that can predict how cells respond to genetic or chemical changes. These models hold the promise for transforming drug discovery by accelerating target identification, optimizing candidate compounds, and reducing the cost of drug development. We spoke to ARC Institute's core investigator, Hani Goodarzi, Ultima Genomics CEO Gilad Almogy, and 10x Genomics CEO Serge Saxonov about how the Atlas will serve academic and industry researchers developing therapies, how it'll help unravel disease mechanisms at a cellular level, and provide broad access to advanced cellular analytics. Hani, Gilad, Serge, thanks for joining us.
Thanks for having us.
Great to be here.
We're going to talk about the ARC Virtual Cell Atlas, the partnership between the ARC Institute, Ultima Genomics, and 10x Genomics, and how this work has the potential to transform drug development and disease research. Hani, let's start with the ARC Institute, which is a nonprofit biomedical research organization in Palo Alto, California. Can you explain what its mission is?
Absolutely. So, in my view, ARC is basically an ongoing experiment by our founders, Patrick Collison, Silvana Connerman, and Patrick Shu. We're basically running this experiment live to understand how best to enable scientific discovery and basically unblock scientists and scientific progress. And I think ultimately the idea is that how can we go beyond this how our current approach to science and science funding, which is, every project has to be quantized into a 4 to 5 years duration with 1 or 2 personnel working on the project, and kind of free scientists from that rubric and enable them to think more long term. The way that I would describe it maybe is that, where is the equivalent of Large Hadron Collider of experimental physics in biology? What are the scale of projects that you need sustained expertise and effort cross-disciplinary across a large group of folks to really enable discovery? And those are the kinds of projects that ARC is designed to tackle via its institute initiatives.
What is the ARC Virtual Cell Atlas, and what problems was the Institute seeking to address with its creation?
So going back to your previous question, and as I said, ARC is built to take on kind of grand initiatives, and it was really founded on 2 initial initiatives. Hopefully there will be more in the future. One was Alzheimer's disease initiative as kind of a model of a complex human disease that remains unsolved. And on the other hand, a virtual cell initiative, how to build a kind of a faithful virtual model of a cell, kind of as an AI model. And the Virtual Cell Atlas is kind of designed as a foundation for training such a model. Now, we started this a few months ago. There are 2 initial contributions to the Virtual Cell Atlas, and of course, hopefully the future is going to be much, much more than that. One was SC Base Count, which is an effort by us at ARC to basically take advantage of agentic AI to mine all publicly available data that was basically 10x genomics chemistry, and we reprocessed all of them under the same rubric with the same pipeline. So it's kind of a very harmonized data set across many species, many types of cells out there, and we have very well annotated all of that data. And in parallel to this, Tahoe Therapeutics, which is a platform therapeutics company that, disclaimer, I'm one of the academic co-founders of Tahoe. They have this really multiplexed platform for generating massive amounts of data, single cell data, and they've also generated and contributed the world's largest perturbational single cell data in the form of Tahoe 100M. And they also chose to release their data set as part of our atlas as well. So currently, it's these two major data sets, numbering to above 500,000 or close to 500,000 cells at this point, and it's a continually expanding. Especially the agentic AI component enables us to continue to accrue publicly available data. But as you also pointed out, there is also a partnership, both with 10x and Ultima, to really massively expand on this frontier and really grow the size of the virtual cell atlas by creating perturbational data designed from the ground up to enable this kind of virtual cell modeling.
Single cell sequencing is changing our understanding of cellular biology by providing insights into cellular variation and the characteristics of rare and specialized cells. What challenges have existed around integrating and analyzing this data on a large scale?
Yeah, Daniel. So maybe I'll kind of one thing that I just want to say maybe up front is the sort of the universality of single cell sequencing as an approach to understanding the fundamentals of biology. I think, this is important because it goes to sort of the crux of this initiative and why we feel so enthused about pursuing it and how big a deal we feel it is. Like fundamentally, the cell is the fundamental unit of biology. And when you look at what single cell, the arrival of single cell biology has made happen, and when we talk about single cell sequencing, we're not talking about sequencing one individual cell. Really, the name of the game is scale. It's being able to do huge numbers of individual cells. That's what it's about, and that's what our tools, our technology, kind of first enabled the world to do now some number of years ago. And it spans in terms of the impact. It goes beyond just sort of the way when one thinks about just cellular biology. It is really goes to the heart of molecular biology, tissue biology, and so on. Not a whole lot in biology makes sense outside of the context of cells, fundamentally. So, and then when you look at what has transpired over the past several years, just like massive discoveries across just about every area of biology. The field kind of got kicked off with the discovery of a new specialized cell in the lung, the ionocyte, which turned out to be the cell that carries the cystic fibrosis gene, kind of changing understanding of disease. And since then, across every area of biology, oncology, neurodegeneration, all kinds of drug treatments has been like really central. So just kind of laying the groundwork for how this technology has been really, really prevalent and like hugely enabling across just about every area of biology. And again, the key here is scale and sort of scale across multiple dimensions, but especially numbers of cells. Each one of us has close to 40 trillion cells, and so there's a lot of work to do to analyze. And to a question around sort of the challenges around integrating, analyzing data. First, of course, is just sort of the quantity of it. Presented challenges, especially kind of early on, and building out the right kind of software and tooling and computational infrastructure has been really important to enable processing of those data. And it's always sort of a continuous race because we keep putting out more technology that can do larger and larger scale of experiments, which require more and more compute on the part of researchers. And then the sort of the other element of that is being able to, as you say, like integrating this data across across different labs, across different experiments. And there, the consistency of tools of your experiments becomes really important as well, right? And that's also part of what we've tried to kind of lead the charge on, is getting this technology out there in the world and to be used in a consistent way to generate these large data sets that can ultimately be integrated. But of course, someone also has to do the integration, and Hani was just referring to this really nice work from ARC, is actually kind of they're using agentic AI to scout the world for these data sets and bring them in and integrating them. And I would say the final bit here, right, is kind of setting up the right incentives for the world to share the data, to integrate it. And sometimes that has been more challenging, maybe given just the incentives of individual researchers to pursue their own, kind of specific research goals, whereas there's a huge benefit to be able to kind of bring the data sets together and kind of do science on a much larger scale than was possible before.
Well, Serge, can you give some sense of the technology that you're bringing to this effort, and what was it about this that you wanted to get involved?
Yeah. So as far as the technology is concerned, we started kind of from the very beginning. Our concept was biology is fundamentally really complex, and addressing that complexity. And it's something that's like, it's a kind of a tried statement, but often said, but I think people still kind of underappreciate fundamentally the complexity, evidently, and depreciate the complexity of biology. And to address the complexity, you need technologies that can measure things at scale, very, very large numbers. And so when I talk about the complexity, I'm talking about the number of cells, all the different cell types that exist, all the different cell states that exist, and all the different sort of molecular species that exist in any given cell, and how they interact within the cell and also between cells. Right. And so to kind of address that complexity, our contention is you need to bring forth tools, technologies that can measure all the complexity at very, very large scale and really high resolution. And so the crux of our of the initial technology that we developed for single cell sequencing called Chromium, the Chromium GEM platform, was to be able to essentially miniaturize your kind of experiment into individual droplets, and then within each droplet have special kind of barcodes, oligonucleotide barcodes, that allow you to now run massive numbers of reactions in parallel. So thousands, hundreds of thousands of millions. And in a particular, and that allows you, if you imagine individual cells within each droplet, that then now you can sort of analyze, gives you a huge, a lot of power to measure lots of things, really, really, in a really, really scalable manner. And in particular for what we're doing here with ARC, we developed this technology called GEMX Flex, enables processing of millions of cells while maintaining that individual single cell resolution. Each cell is encoded with the different CRISPR guide to kind of affect the expression of a particular gene. That's the perturbation, and then our technology captures each cell in its own kind of reaction compartment in the droplet, barcodes all the sort of expression of RNA molecules in there with a specific special identifier. And then as you kind of sequence the contents of all those cells, now you can determine with really high fidelity, really high accuracy and sensitivity, which CRISPR guides, which perturbations went into that cell, and also measure the effects of that perturbation, the full transcriptome of that cell. And this kind of technology sits on many generations of improvements that we have made over the years, again to drive really high scalability, robustness, quality, sensitivity. And then we're really excited to the second part of the question of being able to partner with ARC on this effort, because the vision here aligns perfectly with ours, kind of driving toward the fundamentally accelerating the understanding, the mastery of biology to understand how things work, to understand to pull out sort of the fundamental cellular processes that drive biology, and being able to to measure, kind of extract causality, like what is it that what changes do you need to make to move states, cellular states from one state to another.
Gilad, what technology is Ultima bringing to this effort, and what is it enabling the virtual cell atlas to do that it might not otherwise be able to do, or to do better?
Ultima's premise is that biology will need a lot more raw data, a lot more information. Genomics is a form of that information, whether it's reading DNA or in this case reading RNA. You need access to a lot more information. When we set out to start the company, you could say, well, the human genome has been sequenced over 20 years ago, 3 billion bases or so. That's very doable today. Sequence every person once, solve problem. But I think, as Serge was saying earlier, we are not. Nominally, we have 37 trillion, 40 trillion copies of that genome we inherited. But that's a highly simplistic view. Those 40 trillion cells or so, not only they all start with the same genome, but it evolved. It's mutated. They carry different mutations. It's called methylated. It's got epigenetic signatures. It's folded in a different way, expresses a different way. And in the end, that's what causes the cells to be differentiated. Cells make different RNA, and RNA makes protein. And with this effort here is to use, A, perturb the cells, and B, use the RNA as a readout for what happened to the cells. So we're the readout. And we, from day one, had the premise that you need a lot more readout. If you just need to sequence every human once, solve problem. But if you need to take ultra large scale experiments, such as to train AI, so perturb-seq, where you're perturbing potentially 10,000 genes plus, and you're doing that on many cell types, so populations, so very quickly the numbers add up. And you're talking hundreds of millions of cells and billions of cells, and each cell makes tens of thousands of transcripts of RNA. So the numbers really start adding up really, really quickly. So what we are is a pure play data generator, ultra high throughput, low cost reader of DNA. And our contribution here, the founding contribution here, is to make this economically viable. So take the numbers that you need to actually make an impact in single cell, make those numbers achievable. That's one. That's probably the core. And then there's a lot of collaboration with folks at 10x, with the folks at the ARC Institute, which is okay. It's a lot of data handled. And there's a lot of, this gets into the weeds here, but is it just raw sequencing data, or can you pre-process it, trim it, manipulate it in a way that makes the upstream machine learning that comes after us? We don't do the machine learning and the training. That's done by the scientists. It's done by ARC. But we help the data funnel come out much more smoothly in collaboration with the two folks here on the call.
This is a free-to-use public domain resource. Who can access the Cell Atlas, and how would a researcher use it?
Very good question. So as you said, this is publicly available, and as we add more data to it, those data sets will also be publicly available. It isn't just easy access, it's also well maintained and well annotated. As I mentioned, we already have 2 data sets as part of our virtual cell atlas, which was released a few months ago. It's SC base count and Tahoe 100M. And as we process and generate more data, those data sets will also be added over time to the virtual cell atlas. And more importantly, as Gilad mentioned, at ARC, we also have a virtual cell initiative. So we are not just only releasing the data, but we are also training models on top of this data that we are releasing as well. For example, we just recently published our first virtual cell model called State, which was trained on SC base count and Tahoe, basically our virtual cell atlas currently. And it is really the first of its kind model that beats the simple baselines that have been proven hard to beat so far. And we are going to be on a kind of regular cadence, training and releasing these data sets, these models. And more importantly, we are also not only releasing the model, we are also releasing the training code, kind of the evaluation metrics and all that. And in parallel to all of this effort, we are also running the Virtual Cell Challenge, which is basically a competition designed to enable others to basically compete in an equal footing and be able to compare and contrast different strategies in designing models, model architectures, training strategies, and all that to see that who can we learn lessons from, and how can we incorporate these learnings into better and better models over time.
I do want to talk about the challenge, but before we do, can you give some sense of the data that sits within the atlas in terms of what it includes, the sources of the data, and you mentioned earlier using agentic AI to build it. So is there programs constantly scouring the available public domain databases to add to this?
Exactly. I mean, as I mentioned, there's currently two data sets served as part of our Virtual Cell Atlas. One is SC base count, which, as you mentioned, there is an agent called SR agent that is continuously searching for new additions. And we have already processed the old additions, so we're looking at new additions. And specifically, we first focus on the 10x genomics chemistries, but hopefully we will expand that in the future as well. But basically, the agent identifies new single cell sequencing data sets and then pass them on to basically a downstream pipeline that is uniformly processing all of this data. So we are kind of removing all the technical variations that kind of independent analysis of these different data sets would have created. So we have created this very uniform data set that is quite enabling for training these kinds of models. And in parallel to this, as I mentioned, Tau Therapeutics has also generated and released their largest 100 million single cell data as part of our atlas. And their data set is also very unique. It's the largest data set. There's almost no batch effects in it. It's all uniformly processed and handled. So these are quite enabling in training current models. And I think without them, basically our state model would not have been possible. And again, as part of this partnership with 10x and Ultima, we are also going to continue to generate perturbative data, specifically both genetic perturbations and genetic times chemical perturbation. So basically continue to perturb cells and put them in new environments and understand how these perturbations impact the gene expression landscape of the cell, the state of the cell, and the function of the cell, and be able to learn from that and enable a model to learn from these changes.
What's known about its ability to adequately predict a drug or genetic change and the way that might impact a cell?
Very good question. So this is basically goes to another question around not only how do you build these models, but also how do you evaluate these models, right? So, and what are the metrics for success? So at ARC, we have this basically project called Cell Eval that we have published the first version of it. It's basically created to pretty much answer this question of, at this point in time, these are the metrics that we care about. The way that we are approaching the problem at this point around like what a virtual cell means is a model that can predict the impact of genetic or chemical perturbations, right? And so the cell eval is basically made up of metrics that would evaluate this. And the way that we are setting it up now is what we call a context generalization approach. Basically, the idea being that if we take a data set like SC base count and Tahoe, and Tau contains 1,100 perturbations. So the model will be trained on 1,100 perturbations across 50 cell lines. Now, if I show tens or maybe a couple of hundred perturbations to my model from a new cell line that the model hasn't seen before, how well would it be able to predict the effect of all the other perturbations that we haven't seen? So this is primarily how we are evaluating the performance of our models currently. And of course, as we make better and better, these performance metrics are also going to evolve as well. Now, how does it come into drug discovery? Of course, if you know the impact of genetic perturbations and you can predict which genes do I have to perturb to make a cell that is diseased look like a healthy cell, that gene is my target. So that is a gene that I have to go after. Or similarly, if I can find a drug that would take my cell from a disease state and make it look like a healthy cell, that is the drug that I want. So that's basically how these virtual cell models very quickly get plugged into drug discovery and kind of human application.
Is there anything that could be said about how this might ultimately impact either drug discovery or preclinical development in terms of speed or cost?
So, Hani, continue. I was just going to add maybe a little bit in a sense. We obviously have a lot of exposure to kind of what people are interested in terms of our customers, and a lot of the biotechs and pharmaceutical companies are very keen on exploring these approaches. It's obviously still early, but there's a lot of interest. Just kind of to support what.
So one thing I wanted to add very quickly is that ultimately in biology, what we care about is function. But function is not easy to measure, and it's not very scalable measurements of it. So what we do in biology is we try to find proxies that we can generate lots of data around and be able to model effectively. For example, in the landscape of proteins, there is a very strong structure-function relationship. So two proteins that have the same structure, not even the same sequence, but that have the same structure, likely do the same function. Right now, in the context of cell function, this was very difficult for until very recently. Again, thanks to the pioneering work from Serge and other pioneers in the field, we have now created this idea of a cell-state function relationships for cells. That cells that are in the same state are probably doing the same thing. For example, neurons that have the same cell state as measured by their gene expression landscape via these single cell transcriptomic technologies will share the same functions, carry the same functions. Same applies to any other cell in the body. So that's why a lot of these large scale data generation efforts become useful, because you can actually integrate and compare and contrast the state of the cells, and via this comparison of states also learn something about the function or misfunction of the cells in the context of disease.
You talked about the challenge earlier. This is a global virtual cell challenge that's designed to accelerate the development of AI models capable of simulating cellular behavior. How does the challenge work?
Very good question. I would say we have tried to learn lessons from, again, pioneers in the field. For example, when it comes to protein structure modeling, which was one of the holy grails of much of the past 2 decades in AI and machine learning applied to biology, which basically culminated in Nobel Prizes that you saw earlier. There's one of the big enablers in that field was this challenge called CASP, Critical Assessment of Structure Prediction, and which was basically an annual competition to kind of compare and contrast models and how well they were performing in terms of structure prediction. Now, kind of learning the lessons from that, we realized that we needed something similar in the context of virtual cells. Why? Because folks are developing their own models, training their own models, coming up with their own evaluations, and everyone is almost the hero of their own stories in some sense, right? Everyone does really well in kind of tasks that they benchmark their models in. So the comparisons on equal footing is actually quite hard. So we at ARC decided that we're actually very well positioned to solve for this problem by creating the virtual cell challenge, where we are basically committing to generating a data set every year dedicated to asking the question of which approach out there is actually performing best with a brand new data set that no one has seen before. And for this year's data set, we picked about 300 genes in a human embryonic stem cell called H1 cells, and we generated this really high quality data set. We actually just today released a blog post about kind of all the details of that and all the thought and process that went into selecting the cells, selecting the genes, and selecting the chemistry. Again, thanks to Serge's flex chemistry, we have generated this data. And we basically gave half of this data as training to basically for the competition. So all the participants have access to half of this data, and they are using that for training. And then they are actually there's a live leaderboard with a fraction of the genes. We'll see who is performing well, kind of live, and then finally the final week of the competition, there is going to be a test set that is completely held out, and we will basically announce winners at that point. And there are cash prizes and equivalent prizes, basically enabled by some of our sponsors, 10x included.
I'd like to ask each of you to weigh in on where do you see us going in the next 10 years with this technology, and where do you ultimately see it having the most significant impact? Gilad, do you want to start?
So I think the challenge people have with AI models is that it's incredibly difficult to predict what scope helps you develop the model and how generalizable the model is at each scope. So I think from the data that's been generated that was just mentioned here, there'll be a lot of learning of how much is needed. Biology is complex. Cell interactions are very complex. Cells by themselves are complex. When you start putting them in tissues and organs, become even more. So I believe there'll be a massive amount of scaling, and it won't be 6 months from now, 12, 18 months from now, the holy grail moment, and we have a model that describes every cell in the universe. I think you'll have models in the short term that have some value, and we have some wins. We have some better drug discovery, better fundamental understanding of some phenomena in biology. But the road is long, and I think over the next decade we'll keep seeing improvements again. More cell types, more generalizable models, cells in their environment, more complex environments. So more and more on the cell type. I think there's other places beyond, cells also live in our bodies and we can't always access them. Some we can, like the blood cells. And I think you'll see a lot of like folks developing models on cells actually derived from humans, not from not for perturbation per se. And there's also going to be information in DNA collected from cells that break up, circulating free DNA. So I think there's going to be a lot of data sources, larger and larger experiments, more perturbation types, cells from humans, and information from cells that are broken up. All of these are going to be driving over the next decade.
Serge?
I mean, I agree with Gilad's kind of framing. There's a lot of progress to be made here along multiple axes. Kind of my view this, I think clearly, like we're just at the very beginning, at the very start. And I do, by the way, I just want to emphasize how much I like the approach that ARC is taking here, which is sort of inspired by what has been done in the protein structure world and generally in AI kind of progress world, where these kind of foundational data sets and benchmarks are critical for enabling progress in the field. And it's really good for us to kind of lay that foundation now. But it is just the beginning. We keep we talk about scale, and I think there's should be huge returns to measuring things at larger and larger scale. Just again because biology is complex and there's just so much to kind of to model. Also, starting with expression, you kind of imagine there will be other modalities that we'll want to bring into the equation. Certainly, proteins, chromatin states. Right. And many different, cell states and tissues and types that we'll want to interrogate with time, and more and more combinations of all the different perturbations to kind of to build richer and richer models of biology. And so I think right now we're just again on kind of at the very early days, but as we progress, and as especially on a scale of 10 years, I think like pretty insane amounts of progress are likely to be made. And as far as impact is concerned, I kind of in my mind there's sort of three buckets. Right, there's the fundamental understanding of biology, like just understanding of kind of the basic science of it, and you can imagine the future where if you run an experiment, a grad student or something runs an experiment and they have a question that's like, what is likely an explanation for what I'm seeing? they can refer to these to a virtual cell model that will give it an explanation. Chances are, for example, these are the genes that got turned on in these particular cell states, and that's what you're seeing here, and how to help interpret the results. The drug development sort of paradigm is likely to change significantly when you can go from kind of a trial and error, throwing darts at the wall, in terms of choosing gene targets, which is largely what the world is like today, to where you can have very, very strong priors and be very deliberate around which drug targets to go after for a given condition. And I think, given like sort of the explosion, the progress we're making on different drug modalities, I think this is the key sort of unlock now for drug development, and I think these models, virtual cell models, will be the critical element of unlocking target discovery. And then sort of in diagnostics in the healthcare realm, if you've got a doctor looking, making ultimately some kind of a rich multi-omic measurement of their patient's tissue or blood, and then trying to reason about what is likely going on with that patient, what kind of a disease and is happening there, and what kind of treatment would likely shift, that disease into a healthy state, they would likely be referring to some version of a virtual cell model, together with other sort of likely AI approaches to give them guidance. So I do think like we're very early at this point, or at the beginning, but the kind of the directionally, it's pretty clear where we need to go, and it's very exciting what the next 10 years are going to be like.
And Hani, I'll give you the last word.
I think, it's difficult to predict the future. I would say there are a couple of directions that I would certainly think it is helpful to think about. One is multimodal data, right? As I mentioned, we have been talking about the importance of single cell transcriptomics, but there's more to cells than just RNA, and being able to capture at a similar scale other modalities, for example, protein levels, metabolism, kind of even functional aspects of the biology of the cell, would go a long way in teaching the models how to kind of reason around biology and the impact of perturbations. So I think that would be kind of a very clear direction that we need to think about. We also kind of need to think about the type of concerted efforts that we're describing here. We have to be very deliberate about the next stages of how we think about AI and its application in biology. AI, there's no question that AI is transformative. We have seen impacts of it out there, but the reality is that internet was not built so that it gives training data for AI, right? Or the protein data bank was not built for training data for structure prediction models, right? So we got lucky in some sense to have had infrastructures that then could become a foundation for really amazing AI models. But now in biology, much of biology is data starved. So we have to be very deliberate and think about what kind of data we want to generate, how we want to generate it, and really understand the scale that is needed, and bring together the type of experts and parties involved that are needed to actually really enable and realize that vision.
Arc Institute core investigator Hani Goodarzi, Ultima CEO Gilad Almogy, and 10x Genomics CEO Serge Saxonov. Hani, Gilad, Serge, thank you all for your time today.
Thank you for having us.
Thank you. Great.
Thank you very much.
Thanks for listening. The Bio Report is a production of the Levine Media Group. To automatically download this podcast each week, subscribe to our RSS feed or through iTunes or other podcast manager. To join our mailing list, go to levinemediagroup.com. We'd love to hear from you. If you want to drop us a line or are interested in sponsoring this podcast, send email to danny@levinemediagroup.com. Special thanks to Jonah Levine, who composed our theme music, and the Jonah Levine Collective, which performs it.